Catherine Field is our first speaker. She's a professor in the Department of Agricultural, Food and Nutritional Sciences at U of A. Part of her research program is funded by AI Bio. And she is drawing these interesting connections that I suggested before between crops, the edible oils that those crops produce, and more effective chemotherapy at the other end of that. So, Catherine, welcome.
Thank you, Jay, and thank you, Alberta Innovates, for inviting me here to talk about something that's very close to my heart. So breast cancer is one of the most common female cancers, and that 1 in 9 North American women will be diagnosed over their lifetime with this disease. And despite considerable advances in the detection and the treatment of this disease, last year in Alberta, 23,000 women were diagnosed and 5,000 died from breast cancer. So clearly there are further advances in scientific knowledge that we need to improve the treatment and extend the lives of women who are afflicted with this disease. So there's very convincing population health studies that have demonstrated that high intakes of fatty fish, which are rich in the long-chain omega-3 fatty acids, are consistently associated with a lower risk for breast cancer. In animal models, feeding fish oils will prevent, reduce the growth of breast cancer tumors. So I started in this area over 10 years ago when we were working as a nutritional immunologist I wanted to study what would happen when you feed long-chain omega-3 fatty acids to an animal that had a breast cancer tumor. So we got some mice and we implanted them with a tumor. And we fed them 2 diets, a low omega-3 and a high omega-3 diet. Well, what happened? Well, the tumors wouldn't grow. They actually shrunk. And we kept trying this, and I thought, well, you know, what I really need to do is figure out what's going on. Maybe we have something that could be used in the treatment. So we've been working with a number of models of breast cancer, and we started with cell culture models where we get human breast cancer cells. And what we do is we grow them in different media and we put in different amounts of fatty acids. So across the x-axis here, we have different amounts of docosahexaenoic acid, or DHA. These would be ranges where we would find if you consumed a lot of omega-3 fatty acids or took a supplement in your blood. When we grow cells, if they are not reduced in growth, they grow fine, you get a straight line. And that's what happens when we put a non-tumorigenic, non-malignant human breast cancer cell line, so healthy cells. But when we put any of the 3 commonly studied breast cancer tumor cell lines that represent the whole range of cancer tumors that women are diagnosed with, they very quickly die. So what is going on? And really that's what we spent our time trying to figure out what's going on. Well, when we work with these omega-3 fatty acids, we're really talking about a whole family of fatty acids. The most common being alpha-linolenic acid that can be found in various amounts in our diet, canola and flax being the largest without very much coming from the corn or the soybean oil. Well, these really are important because they're elongated in the body to these long-chain fatty acids, EPA and DHA, and really these are the ones that are the essential fatty acids for function and have the biological effect. Well, the problem is this enzyme here at the very first one in the step, and that this is rate limiting. And yes, if you consume ALA, it will be converted to this, but the amount that can be converted to these amounts is limited by this enzyme. So how do we get around this? And this is really where the agricultural connection comes in. Well, the first obvious way is, well, we could just consume these directly in our diet, and yes, we do when we consume the fatty fish, or we take supplements that the primary ones being marine origin. We've got some new plankton sources from krill, and we've got the single-cell organisms. So what's going on in this, back to this tumor model? Well, there's 2 ways that the tumor cells could be affected by these omega-3 fatty acids. The first is through a process called apoptosis, and this is really a cell suicide process where the cells kill themselves. And this is controlled by a series of receptors called death receptors. Or we could decrease the growth or replication. Again, this is controlled by a series of receptors called growth receptors. And this is really where the focus of our research has been over the last 10 years. So we've done a series of experiments where we've looked very closely and showed, yes, DHA and EPA both increase apoptosis and cell suicide by increasing the death receptors in the plasma membrane and increasing genes and proteins that are involved in apoptosis. And yes, it decreases replication by decreasing growth receptors in the genes and proteins that regulate the growth receptors. So it's acting on both sides of this pathway. But again, only in the malignant cells. Healthy cells actually just grow better. But when we want to move into cancer, we do have effective treatments for cancer. And doxorubicin is probably the most commonly used cancer drug that's used in the treatment of almost all forms of breast cancer. And what's really interesting about this form of treatment is that the mechanisms in which that it affects the cancer cells are very similar to what we found for the omega-3 fatty acids. So in 2008, we proposed in a paper that maybe we could use these fatty acids to help improve the efficacy of our breast cancer treatments. And yes, it works. This is a cell that looks at cell survival, how well cells grow. And this is our control fatty acid treatment where the cells grow really well. Doxorubicin decreases the growth of cells, and that's what it's supposed to do. DHA does as well. When we treat the cells with DHA before we give the doxorubicin, the cells grow even less well, which would be a good thing for treatment of cancer. How does it do it? Well, for many years of experimental work, we focused in on what we think the key mechanism is. And this is the expression of a death receptor in the plasma membrane called CD95, or sometimes the FAST receptor. In cells that have been incubated in control fatty acids, not very many express the death receptor. When we incubate them with DHA, more cells express the death receptor. If we incubate them just with the doxorubicin, even more express the death receptor. When we treat them first with DHA, remove them from the media, then treat them with doxorubicin, even a higher expression. And that's one of the key mechanisms we think, and now we're doing studies to block that, and it looks like this is the mechanism in which they act. So we have ongoing research now in this area, and we are exploring a plant source of EPA and DHA from canola, because as a nutritionist, the translation of this to the human is not necessarily that we need to use even more fish oil, because there are some environmental concerns about that. The single-cell sources are quite expensive, and what about a way that would be very acceptable and easy to get into the diet would be through oils. And there is now a canola that is in the pre-market stage that does contain EPA and DHA similar to what you would find in fish oil. We're also moving to an animal model, and I'll talk a little more about that, where we can implant a human breast cancer tumor in an animal and study the dietary effects, which would be the sort of pre-clinical type of experiment. So what's the other approach in trying to get more of this EPA and DHA with this rate-limiting enzyme? Well, what if we looked at a fatty acid that bypassed that came and passed that didn't need the enzyme to be converted? And this is where our work other end of our work is focused on this